Hello, everyone. My name is Sarah Hadyniak from the Johnston Lab at Johns Hopkins University, and I'm excited to talk to you about my work on human cone cell development. I'd like to thank the organizers for giving me the opportunity to talk to you today. The goal of my research is to understand a longstanding question in retinal biology on the developmental order of green and red cones. These are the cells important for visual cues that help you differentiate the colors in your world, or in absence of any cone cell subtype yields to the loss of ability to compare hues. Deuteranopia is the loss of green detecting cone photoreceptors, where one would see something like this. Humans have trichromatic color vision conferred by blue, green, and red cone cells, which are responsible for color and daytime vision. We also have rod photoreceptors responsible for low light and motion detection. This is an adaptive optics image of a human macula showing the three cone cell subtypes. This type of image is captured in living subjects. If you look throughout the animal kingdom, I hope you can appreciate the diversity in cone cell subtypes in some of our most favorite model organisms here. Humans are the only ones, however, with trichromatic color vision. Mice have rods, blue and green cones. Red cones are exclusive to humans, old, and some New World primates among mammals, making this question difficult to study in a model system. Additional challenges are that green and red cones differ by their respective opsin proteins only, a GPCR that is 96% similar. These black circles indicate the amino acid differences between the two proteins, most of which lie interior to the membrane. Therefore, antibodies are not specific for red or green opsin. This is a flat mount view of a human retina where we have labeled the photoreceptors here. And as you can see, green and red cones are co-labeled together in green. The Johnston Lab therefore uses human retinal organoids to study questions about human cone cell development. This is a day 200 organoid, which is also why I am almost a sixth-year graduate student and still going, but the outside of these organoids contain the photoreceptor layer reaching into culture. If we stain for photoreceptors, we find our retinal organoids have all green and red cones in green, blue cones in cyan, and rods in fuchsia that look like here. There are two main models for green and red cone cell development in the field, the stochastic model and the temporal model. The prevailing model in the field is the stochastic model, where green and red cones choose their fate from a pool of retinal progenitor cells, which give rise to all of the cell types in the retina. In this model, blue cones are generated first temporally, and then green and red cones choose their fate stochastically. Red and green opsin lie in an opsin array on the X chromosome, where a locus control region, or LCR, is an enhancer required for the expression of both red and green opsin. It has been hypothesized by the Nathan's lab here at Hopkins that cells randomly choose one opsin or the other via a distance-dependent DNA looping mechanism. A second model in the field is the temporal model, where all three cones subtypes arise in distinct temporal windows. The temporal model is suggested by several imaging techniques looking at the far periphery of the eye and qPCR studies on relative opsin abundance. Additionally, it is important to note that these models may not be mutually exclusive, but for the rest of the talk, I will show you data that supports the temporal model for cone cell generation. So I set out to provide evidence for these models. So the first question we wanted to ask was quite simple. How are green and red opsin, the only known transcript level difference between green and red cones, expressed during human development? And this talk is summarized in a paper in prep that I should be submitting very soon. So if you align the mRNA sequences, they are 98% similar, the same length, with 20 differential SNPs between green and red opsins shown by these red bars. Previously, aligning sequence reads to these opsins was extremely difficult due to the redundancies of the sequences. So I teamed up with Boris, a graduate student, and the Taylor lab at Hopkins to address alignment challenges. Using a previously published human embryonic and fetal data set by the Swaroop and Ray labs, we looked at each opsin by normalized pileup counts. That is, each time we saw a SNP present in any given read and the total, we divided that by the total read count, and we assigned it to the appropriate opsin. So on this diagram, you have the 20 differential SNPs for green opsin, 5' to 3', and the 20 differential SNPs for red, 5' to 3', and developmental time is increasing on the x-axis. So what do we see? For green opsin, we see onset starting at day 115 and increasing. For red opsin, we see nothing during these time points. If this were a stochastic model, we would expect to see both onset of both opsins at the same time, but we only see green onset, which implicates temporal inputs rather than stochastic in the system. If you look at human adult retinas, where we have 3 individuals listed from a previously published dataset, what I hope you can appreciate is we have expression of both green and red opsin. In fact, if you compare within individuals, you see higher red levels, which matches an average higher red cone ratio to green cone ratio, which is something we'll come back to later in the talk. So we can conclude that both our analysis works for both differentiating the opsins and that green opsin onset is expressed first in human development. We have access to time points approximately halfway through human development, but we were interested in gathering clues to determine when red opsin onset occurs. Rhesus macaque monkeys have trichromatic color vision, and with samples generously donated by the Latour lab, we were able to gain insights into the later half of macaque development. Rhesus macaque opsin sequences have 24 differential SNPs between each, and using the same analysis for macaque, we see no opsin expression of either green or red opsin at day 90. At day 110, while green opsin is expressed at higher levels compared to red, both opsins are on at day 110. And as developmental time continues, we see red opsin levels increasing comparative to green. What we can conclude from this is that green opsin is expressed at higher levels at onset and that red opsin does turn on during fetal development in macaque. So the next question we asked was, what happens in our retinal organoids? Here we have the developing human and the adult retina data sets again. And if we look at human retinal organoids, we see robust expression of green opsin from day 158 of differentiation on to day 250. And we see no expression of red opsin and no red cones. We can therefore conclude that green opsin is also expressed first in retinal organoid culture. But you might be sitting there thinking, where are the red cones? When we found out the data suggesting organoids only had green cones, it was a bad day, week, month. But it became a silver lining, actually, because we were able to manipulate the system and ask the question of what factors specify red cones. And first, let me tell you where we found some clues in order how to do so. What is really important to understand is that the ratio of red to green cones is highly variable in the color vision normal population, from 1 to 3, 1 to 1, and 17 to 1. And all of these individuals, again, see normally. In collaboration with the Rhesus group, they determined the % red ratios and the DNA sequences from 738 human males. And they have a nice normal distribution, as you can see here. Males were used because the opsin locus, again, is on the X chromosome, and therefore they only have 1 copy. We asked the question, what changes in DNA sequence associate with changes in red to green ratios? We sequenced 20 relevant retinal patterning genes and the opsin locus to do so. These are the data on this Manhattan plot. On the x-axis is the 20 relevant retinal patterning genes and the opsin locus. And we've had one significant variant in the non-coding region of NR2F2, which is a haplotype where changes in one or both of the flanking bases is associated with changes in the % red to green ratio. This variant is in an antisense RNA to NR2F2, which is part of the thyroid hormone family of nuclear receptors activated by its ligand, retinoic acid. Retinoic acid is also important in zebrafish cone development. Zebrafish have 2 unique long wavelength detecting opsins and a shared enhancer element called a locus activating region, or LAR. In early development, the further away opsin is expressed, and in late development, retinoic acid regulates the change from LW2 to LW1. In zebrafish, the zebrafish locus bears resemblance to the human locus, though independently evolved, where I've shown that the further away opsin is expressed first. I therefore hypothesized that retinoic acid could be playing a role in the generation of red cones. But first, we needed a way to visualize red and green opsin. As I previously mentioned, we didn't have a way to differentiate the two, and a concerted effort to tag the locus was unsuccessful. I turned to an in situ approach that differentiated between these 3 SNPs in green and red opsin, the probes binding these regions highlighted here. Using HEC293 cells probing for both green and red opsin mRNA, the negative control we see nothing. and the green opsin signal in the green opsin transfected cells, red opsin signal in the red opsin transfected cells, either green or red signal when these populations of cells are mixed, and a dark purple stain when the plasmids were co-transfected. This was the first time that anyone has been able to visualize green and red opsins in fixed tissue, and so I was excited to use this technology going forward in human retinas and organoids. This is a section of a human retina with a photoreceptor layer on top stained for rods, green and red opsin in green, and blue opsin in blue. And we can visualize green and red opsin for the first time in sectioned human tissue, as you can see here. So this brings us to the question that I wanted to ask: does retinoic acid signaling specify cone fate? I looked to alter the windows of retinoic acid signaling in our retinal organoids and test if we could generate red cones in our system. I was especially interested in the day 130 to 150 window because this window was critical for green opsin onset. In our original protocol, we added retinoic acid from days 43 to 130 and produced all green cones. This agrees with our RNA sequencing data. Retinoic acid was stopped at day 130 because it was previously thought to be required for the generation of a variety of cell types in the retina, but not their maintenance. If I extend the window of retinoic acid signaling 20 days, we see little change and mostly green cones. If I pulse retinoic acid from the 130 to 150 day window, we start to see the presence of red cones with a minority of co-expressing cones. And finally, if we add retinoic acid late from days 130 to 200, we see organoids with nearly entirely red cones. And I can quantify this, where we see the addition of retinoic acid in later time windows increases red opsin. We still see high variability in the short pulse between days 130 to 150, but we can conclude that retinoic acid signaling late is sufficient to generate red cones as indicative of the 130 to 200 day addition, and a significant difference between this and the early treatment of retinoic acid. I therefore conclude that I've shown evidence towards the temporal generation of cone cell subtypes, where we have known that blue cones are generated first, followed by green cones, and finally red cones. And I've shown that retinoic acid plays a role in the late generation of red cones. To conclude, I have shown that green opsin precedes red opsin onset, that NR2F2, a receptor for retinoic acid, is associated with changes in red to green ratios in humans, and that retinoic acid signaling induces red cones in organoids. And with that, I must thank the individuals who have supported my work, my lab, my funding sources, collaborators, and be sure to check out the Johnson Lab here at SDB, and feel free to reach out to me with any questions. Thank you.